Before we jump in, here's the takeaway for today's deep dive. The human nervous system, that incredibly complex network running your entire life, is not a static object. It is a history of timing.
It really is.
It's a story of a flat sheet rolling into a tube, of cells knowing exactly when to let go and migrate, and of a zipper that has to close within a specific 24-hour window or everything changes.
Exactly. If you understand the timing, you understand the anatomy. And if you understand the anatomy, you understand the pathology.
Welcome back to The Deep Dive. Today we are tackling wiring the body. We are looking at the embryology of the nervous system. And I have to say, looking at the stack of research we have for this one, this is heavy stuff.
It is heavy, but it's foundational. You literally cannot be a competent physician, especially, a neurologist or a surgeon, without grasping this.
And to help us navigate this, we aren't just winging it. We're synthesizing three of the absolute titans in the field.
The holy trinity of embryology, if you will.
We have Langman's medical embryology, which is going to be our guide for the molecular heavy lifting, the signaling pathways, the genetics.
All the really nitty-gritty stuff.
And we have Moore & Persaud's The Developing Human, which is just brilliant for the clinical grounding and giving us those real-world applications.
And finally, I.B. Singh's human embryology, which is, well, it's the student's best friend. It breaks things down into clear, exam-oriented patterns and lists.
Now, a quick disclaimer before we start. We are going deep, but we're just guides. This conversation is an interpretation and a synthesis. It is not a replacement for cracking open the books.
Absolutely not. Think of this deep dive as your ready reckoner or your revision aid. We want to give you the mental framework, the coat hanger, so to speak, so that when you go back and read Langman or Moore, the details have somewhere to hang.
So grab your coffee, maybe a notebook, and let's wire the body. The mission today is to figure out how we get from a simple, flat slipper of cells to the structure that allows you to think, feel, and listen to this conversation.
It's a process that's almost more like origami than biology.
Okay, let's start at the beginning then. Section 1, the blueprint. You mentioned a slipper.
I did. So set the scene in your mind. We're in the 3rd week of development. If you look at the embryo from the top, on the back, the dorsal side, there's a thickening of the ectoderm.
No?
This is the neural plate. And it's shaped really like a slipper. It's wide at the top where the head will be and narrow at the bottom where the tail will be.
All right, let's pause there. Usually we talk about the 3 germ layers in the embryo, ectoderm on the outside, mesoderm in the middle, and a germ on the inside.
Correct. And the entire nervous system, all of it, comes from that top layer, the ectoderm. Specifically, a specialized part we call the neuroectoderm.
But Langman makes a really interesting point here about why this plate forms. It doesn't just decide to become a brain on its own.
No, not at all.
It gets bullied into it.
Bullied is one way to put it. It gets inducted. This is a classic example of what you could call embryonic peer pressure. There are structures underneath the ectoderm, specifically the notochord and the paraxial mesoderm, that act as managers.
The middle management.
Exactly. They send chemical signals up to the ectoderm, essentially saying, hey, stop being skin, start being brain.
And this is where the molecular stuff from Langman is it's fascinating. Because it turns out the default setting for these cells is actually to become skin, right?
Precisely. Langman explains that ordinarily the ectoderm is programmed to become epidermis. It receives these constant signals from molecules called BMPs, bone morphogenetic proteins.
So the rule is high BMP equals skin.
Right. But the notochord, that manager sitting underneath, it secretes inhibitors. And these molecules have some of the best names in biology: noggin, cordon, and follistatin.
I love noggin. It's so literal. It helps make your noggin.
It really does. So these molecules, they go up, they find the BMPs, and they just block them. They inhibit the inhibitor of neural formation.
So it's a double negative.
Exactly. By blocking the skin signal, the default programming switches to neural tissue. So low BMP allows that slipper-shaped neural plate to form.
That is wild. So if you didn't have noggin and cordon, you'd essentially just be a ball of skin.
Essentially, yes. You wouldn't form a central nervous system. But once that plate is established, the physical architecture starts to change. This is where we enter the folding stage.
Okay.
I call this the taco stage.
The taco stage. Okay, paint that picture for us.
So the plate lengthens, and then the lateral edges, the sides of the slipper, start to rise up. We call these the neural folds.
Huh?
And because the sides are going up, the middle part gets depressed, forming the neural groove.
So it really does look like a taco shell. Or maybe a hot dog bun.
Yes, exactly. And eventually, those two sides, the folds, they come up, they meet in the middle, and they fuse. This turns the open plate into a closed tube, the neural tube.
And this isn't an instant process, like just clapping your hands together. It's more gradual, more like a zipper.
It is. The fusion begins in the cervical region, basically where your neck is, and then it zips in two directions, cranially towards the head and caudally towards the tail.
But the zipper doesn't close all at once. There are openings left at the top and bottom for a little while.
Right, these are the neuropores, the cranial neuropore at the top and the caudal neuropore at the bottom. And they're open to the amniotic cavity so fluid can flow through. But they have very, very strict deadlines for closing. If they miss the deadline, we have big problems.
Yeah, IB Singh gives us some really good memory anchors for these dates. If you're a student listening, you'll want to write these down. What's the deadline for the top zipper?
The cranial neuropore, the anterior one, closes around day 25. And that's roughly the 18 to 20 summamite stage if you're counting summites.
Day 25. Got it. And the bottom one.
The caudal neuropore closes about 3 days later, around day 27 or 28.
Now, here's where it gets really interesting, and where Moore and Prasad throws a bit of a curveball into the standard textbook explanation. We usually teach it as this smooth single zipper going up and down. But Moore suggests it's not that simple in humans.
This is the 5-site closure hypothesis. Moore notes that the clinical evidence, from looking at birth defects, suggests it might not be a continuous zipper at all.
So not one zipper.
Instead, there might be 5 distinct sites where fusion starts independently and then spreads out from there.
5 separate zippers, all working at once.
Essentially. And this is clinically huge because it helps explain why neural tube defects happen at specific levels. For example, if site 1 fails to close, you might get spina bifida in the lumbar region. If site 2 up near the head fails, you get anencephaly, where the brain doesn't form.
That makes so much more sense. If it was just one zipper, you'd expect the defect to always be in the very end where it failed to close. But since we see holes in different spots, there must be multiple starting points.
Precisely. We're going to get deeper into those defects later, but it really highlights that this isn't just a static anatomy lesson. It's a dynamic race against time. If that zipper jams on day 25, the whole system is compromised.
So we've successfully, hopefully, closed the tube. We have a hollow cylinder running down the back of the embryo. But a hollow tube isn't a spinal cord yet.
No, not even close. Now we have to build the walls. And this leads us to section 2, the architecture of the spinal cord.
IB Singh breaks this down into 3 distinct zones or layers. And I like to think of this as a construction site or maybe like a city planning layout.
It's a great analogy. The wall of the neural tube thickens and organizes itself in a very structured way.
So walk us through the layers, starting from the inside, from the hole in the middle.
Okay, the innermost layer, the one lining the hole, which is the neural canal, is the ventricular zone. It's also called the ependymal layer. This is the factory floor.
The factory floor. Why do you call it that?
Because this is where the cells, the neuroepithelial cells, are just proliferating like crazy. They are dividing and making new cells. That is their only job. Production. Nonstop.
Okay, so once a cell is born on the factory floor, it doesn't just hang around.
No. Once it's born and it stops dividing, it migrates outward. It moves to the next layer, which is the mantle layer or the intermediate zone.
And this is the residential district.
Exactly. The cells migrate here and they differentiate into neurons. They settle down. This mantle layer eventually becomes the gray matter of the spinal cord. This is where all the cell bodies live.
So factory floor, the ventricular zone, makes the cells. They then move out to the suburbs, the mantle layer, to settle down and become neurons. What's the third layer?
The outermost layer is the marginal layer. The neurons in the suburbs, in the mantle layer, they start sending out their long cables, their axons, and these axons extend into this outer rim.
And because axons are often wrapped in myelin, which is fatty and white.
This layer becomes the white matter. It's the information superhighway.
Factory, suburbs, cables, ventricular, mantle, marginal. That structure really holds up. But the spinal cord isn't just a random blob of gray and white matter. It's highly organized. The back of your cord does different things than the front.
And this is the crucial concept of patterning. It's a battle, or maybe a partnership, between 2 plates, the alar plate and the basal plate.
I remember struggling with this in school. I always got them mixed up. Help us out with a memory anchor here.
Okay. Imagine you're looking at a cross-section of that tube. Inside, on the lateral walls, there's a small groove running from top to bottom called the sulcus limitans.
The sulcus limitans.
Think of it as a fence. It divides the tube into a dorsal half, the back, and a ventral half, the front.
Okay, the fence is up.
The dorsal side is the alar plate. Alar comes from the Latin for wing, like a bird's wing on its back. The alar plate is dedicated to sensory processing. afferent signals coming in.
LR equals wing equals back equals sensory.
You've got it. The ventral side, the front, is the basal plate. Basal means base or foundation, the bottom. The basal plate is dedicated to motor function. Efferent signals going out.
Basal equals base equals front equals motor. So LR is for feeling, basal is for doing.
Perfect. And Langman explains the molecular tug of war that sets this whole thing up. It's all about gradients of chemicals.
This goes back to the GPS system for the cells, right? How does a cell know it's in the basal plate and not the LR plate?
It smells the chemicals. Remember the notochord sitting underneath the tube?
Yep.
It secretes a protein called sonic hedgehog, SHH.
Still the best named protein in all of biology.
Without a doubt. So SHH is in a high concentration at the bottom, the ventral side, and it sort of fades out as you go up. A high concentration of SHH tells cells, you are motor neurons.
And at the top, what's the counter signal?
The roof of the tube and the overlying skin, they secrete BMPs and TGF-beta. So a high concentration of BMP tells the cells, you are sensory neurons.
So a cell knows its entire identity simply by where it sits in the tube. If it smells a lot of sonic hedgehog, it becomes a motor neuron. If it smells more BMP, it becomes sensory.
Exactly. Your position determines your destiny. And the cells in the middle, right near that sulcus limitans fence, they get a mix of both signals, and they become interneurons, the connectors.
That is just incredibly elegant. It creates a perfectly organized system without needing a central architect to place every single brick.
It does, but there's a group of cells that doesn't play by these rules. They don't stay in the tube at all. They decide to leave the construction site entirely.
Yes. Section 3, the explorers, the neural crest cells.
Often called the fourth germ layer because they are just so diverse in what they become. These cells appear right at the crests of the neural folds, the very top of the taco shell, if you will.
And when the tube closes?
They detach. They undergo what's called an epithelial to mesenchymal transition. Basically, they let go of their neighbors, they become free agents, and they start migrating throughout the entire body.
And Singh provides a checklist of what these guys become. And it is a bizarrely diverse list. It's not just nerves, is it?
Oh, no, it's everything. First, you have the obvious neural stuff. They form the sensory ganglia, like the dorsal root ganglia that sit just outside the spinal cord.
Okay.
They form the autonomic ganglia, all your sympathetic and parasympathetic nodes. They form the Schwann cells that wrap the peripheral nerves in myelin.
So far, so good. That all makes sense. It's all nervous system related. But then it gets weird.
Right. They form melanocytes, the pigment cells in your skin.
Wait, so the reason I can tan, that's because of cells that started out as part of my nervous system.
Because of neural crest, yes. They also form the adrenal medulla, the center of your adrenal gland that pumps out adrenaline, the fight or flight response.
And this is one that always surprises me. They build the face.
They do. They form much of the skeleton and connective tissue of the face and neck, the odontoblasts that make the dentin in your teeth. Neural crest, the cartilages of your pharyngeal arches. Neural crest.
I think this is a perfect moment for a clinical vignette. Let's imagine a pediatrician seeing a newborn. The baby has a cleft palate, maybe some funny-looking low-set ears.
Okay.
And then they listen to the heart and find a murmur, specifically a defect in the wall dividing the heart chambers.
Classic presentation.
To a layperson, the face and the heart seem totally unrelated, but to an embryologist,
It's the same story. It all points back to the neural crest. Those cells contribute to the facial skeleton, but they also migrate down into the developing heart to help form the septum, the wall that divides the aorta and the pulmonary artery.
so if the migration is lazy or gets stopped for some reason.
You get a package deal of defects, you get the facial anomaly and the heart defect together. This is the basis for things like DeGeorge syndrome or Treacher-Collins syndrome. It's all about those explorers failing to reach their destination on time.
That brings it home. It's not just a random list in IB Singh. It's a roadmap for diagnosis.
Exactly. It connects everything.
So we've built the spinal cord, we've patterned it into motor and sensory, and we've sent the explorers out to build the face and the peripheral nerves. But we haven't talked about the command center yet?
No, we haven't.
Section 4, brain development.
The brain, the big boss. Initially, when the neural tube zips up, the head end just kind of balloons out. We start with 3 primary vesicles, or bubbles.
These are the prosencephalon, which is the forebrain, the mesencephalon, the midbrain, and the rhombencephalon, the hindbrain.
But by week 5, the plot thickens. Those 3 split into 5 secondary vesicles.
Let's run through that split because this maps directly to adult anatomy. It's a key thing to remember.
Okay. The forebrain, the prosencephalon, it divides into 2 parts. The telencephalon, which will become your massive cerebral hemispheres, the wrinkly part of the brain you think of, and the diencephalon, which is buried deep inside, becoming the thalamus, the hypothalamus, and the eye cup.
Okay, forebrain is done. The midbrain.
The midbrain, or mesencephalon, is the stubborn one. It doesn't split. It stays as the mesencephalon.
Easy enough. And the hindbrain.
The rhombencephalon splits into the metencephalon, which becomes the pons and the cerebellum, and the myelencephalon, which becomes the medulla oblongata at the very bottom.
Now, Langman and Moore both emphasize that the brain grows way, way faster than the skull that's trying to enclose it, so it has to fold.
It creates flexures. Think of trying to fit a long, inflating balloon into a small, rigid box. You have to bend it.
We have the cervical flexure between the hindbrain and spinal cord, then the cephalic flexure in the midbrain region. But the most interesting one, the one with the biggest consequence, seems to be the pontine flexure.
The pontine flexure is crucial. It's a reverse bend right between the metencephalon and myelencephalon smack in the middle of the hindbrain. And when it bends backwards, it causes the roof of the hindbrain to stretch out and get really, really thin.
Like stretching a piece of pizza dough.
Exactly. This thinning roof eventually becomes the roof of the fourth ventricle, but it has a major structural consequence that explains the anatomy of the entire brainstem.
Listeners, if you missed the alar versus basal plate discussion earlier, hit rewind for 30 seconds because this is where it pays off.
Okay, so imagine the neural tube is a closed book. Remember our alar-basal conversation?
Right. Basal for motor is at the bottom or front. Alar for sensory is at the top or back.
When the pontine flexure happens and that roof stretches, it's like taking that closed book and opening it flat on a table. The top pages, the dorsal sides, fall out to the lateral edges.
Oh, I see. So the orientation completely changes relative to the midline.
Yes. In the spinal cord, motor is ventral in the front and sensory is dorsal in the back. But in the brainstem, because of this book-opening event, the motor nuclei from the basal plate end up in the medial position, close to the midline. And the sensory nuclei from the alar plate get pushed way out to the lateral position.
That is a fantastic visualization. So if I'm looking at a cross-section of the brainstem and I see a nucleus right in the middle, it's almost certainly motor. If I see one way out on the side, it's sensory.
Precisely. And that holds true for all the cranial nerves that originate there. It's all just geometry. The book opening model is the key to understanding brainstem nuclei without having to just memorize a giant list.
Speaking of position and geometry, let's talk about the spinal cord again for a minute because it doesn't stay put. This is section 5 of our outline, the ascending cord.
Right. This is a race. It's a race between the nerves and the bones. Initially, in the early embryo, around the 3rd month, the spinal cord extends the entire length of the vertebral column.
So at that stage, the spinal cord goes all the way down to the tailbone.
All the way down, but then the vertebral column, the bone, starts growing much faster than the spinal cord, the nerve tissue.
The bone outgrows the nerve.
Exactly. And since the top of the cord is anchored to the brain, this growth differential effectively pulls the end of the spinal cord upward relative to the bony spine. By the time you were born, the tip of the spinal cord is only at the level of the L3 vertebra.
And in a full-grown adult,
it ends even higher, usually at the border of L1 or L2.
This has huge practical implications, doesn't it?
Massive. Think about the spinal nerves. They originate from specific segments of the cord. If the L5 cord segment gets pulled all the way up to L1, but its exit hole, the intervertebral foramen, is way down at the L5 vertebra, well, the nerve has to stretch.
It has to travel a long way down inside the canal just to reach its exit.
Exactly. And this creates a whole bundle of nerve roots hanging off the solid end of the cord that looks exactly like a horse's tail, and that's its name, the cauda equina.
And there's a tether, right? Something holding it down so it doesn't just spring up.
Yes, the filum terminal. It's a delicate little thread of pia mater, the inner covering of the cord, that anchors the very tip of the cord all the way down to the posicix so it doesn't snap up like a window shade.
And for anyone who has ever had a lumbar puncture, a spinal tap, or for any clinician performing one, this anatomy is the absolute safety net.
It is the whole reason we can do it safely. If we need to sample cerebrospinal fluid, the CSF, we insert the needle between the L3-L4 or L4-L5 vertebrae. Why? Because the solid spinal cord ends up at L1-L2. Below that, it's just the horse's tail.
And the nerve roots in the horse's tail are just floating in fluid.
Right. So when the needle goes in, it just pushes the floating nerve roots aside rather than piercing the solid, delicate cord. It's the safe zone. If the cord went all the way down, spinal taps would be incredibly dangerous.
That's a perfect example of embryology dictating day-to-day clinical practice. You can't just know the anatomy. You have to know how it got there to understand the safe zones and the risks.
It is. It absolutely is.
Which brings us to our final section, when it goes wrong. The clinical correlates. We've been talking about the zipper closing the tube. The most common and devastating issues arise when that zipper fails.
These are the neural tube defects, or NTDs, and they are surprisingly common if not prevented.
Let's break them down. I.B. Singh lists a few different types.
The most famous is spina bifida. Now, strictly defined, that just means a failure of the neural arches, the bone, to fuse over the tube. But what really matters is what happens to the stuff under the bone.
And there are degrees of severity here. It's a spectrum.
Right. At the mildest end, you have spina bifida occulta. Occulta means hidden. The bone didn't fuse, but the skin covers it. The spinal cord itself is usually fine. You might just see a little tuft of hair or a dimple on the lower back.
Most people with occulta probably don't even know they have it.
Exactly. But then you get into the cystica types. Spina bifida cystica. This is where things actually protrude out. You can have a meningocele, where just the meninges, the fluid-filled covering, bulge out in a sac.
That sounds bad, but the nerves themselves are still inside the canal.
Usually, yes. The cord is in the right place. But the most severe form is a myelomeningocele.
Myelo meaning the cord itself is in the sac.
Yes. The neural tissue is displaced out into that sac on the baby's back. It's exposed or damaged. And this almost always leads to paralysis, sensory loss, and bladder and bowel issues below the level of the lesion.
And that's if the bottom zipper fails. What if the zipper fails at the top, the cranial neuropore?
Then you get anencephaly, sometimes called meroencephaly. The brain, for all intents and purposes, doesn't form. The skull vault is missing. The developing neural tissue is exposed to the amniotic fluid, and it just degenerates. This is effectively 100% fatal.
Now, Moore and Langman both highlight the prevention story here, which is arguably one of the biggest public health wins of the 20th century.
Folic acid.
It turns out folate, or vitamin B9, is absolutely essential for DNA synthesis and keeping that zipper moving.
The stats are incredible. The fortification of flour and grains with folic acid in many countries reduced the rates of NTDs by about 25%, sometimes up to 70% in some populations. It's a massive victory.
But the timing is the tricky part, and this is the big takeaway for anyone listening who might be planning a family.
Right. Remember the dates. The anterior neuropore closes on day 25. The posterior on day 28.
That is just 4 weeks after conception.
Most women do not even know they are pregnant yet at day 28. That is why the recommendation from every major medical body is for all women of childbearing age to take 400 micrograms of folic acid daily.
Because if you wait until you get the positive pregnancy test,
the zipper has already closed, or it has failed to close. The game is over before you even knew it had started.
That is a very sobering thought. It's all about being prepared for that incredibly early window.
And there are other defects, too, related to the molecular signaling we talked about. Remember SHH sonic hedgehog?
The ventral signal that says be motor.
Well, it also has a critical job in the brain. It helps separate the left and right hemispheres. If SHH signaling goes wrong in the midline, you get a condition called holoprosencephaly.
Holo, meaning whole. The forebrain stays as one sphere.
It fails to split. In severe cases, the eyes confuse, leading to cyclopia, one single eye in the middle of the forehead, or a single nasal chamber. It shows how SHH is crucial not just for motor nerves, but for defining the entire midline of your face and brain.
And finally, one that most people have heard of, hydrocephalus.
Water on the brain. This is often a plumbing issue. We talked about the ventricles, those hollow spaces left over from the original tube. They make fluid, cerebrospinal fluid. That fluid has to circulate and then be reabsorbed.
And the connections between the ventricles are very narrow.
Very narrow. Especially the aqueduct of Sylvius in the midbrain. If that gets blocked, maybe by a genetic defect or an infection or a bleed, the fluid backs up.
Like a clogged drain in a sink.
Exactly. The ventricles upstream expand, pressing on the delicate brain tissue. In a fetus or a newborn, because the skull sutures, the joints between the bones, haven't fused yet, the head can enlarge dramatically to accommodate that pressure.
It's incredible how resilient the system tries to be, even when things go so wrong.
It really is.
So, bringing this all together, we've covered a lot of ground. We started with a slipper.
We rolled it into a taco, zipped it into a tube, and then sank it under the skin.
We watched the notochord shout instructions using Noggin and Sonic Hedgehog to decide what becomes skin and what becomes brain.
We saw the neural crest explorers migrate out to build our faces, our heart valves, and our skin pigment.
And we saw the brain fold and twist to fit into its container, while the spinal cord played a desperate game of catch-up with the growing spine.
It really reinforces the core idea we started with. The nervous system isn't a static thing. It is a history of timing. A delay of just 24 hours in that zipper closing changes a life completely.
It makes you appreciate the incredible precision of it all. We often take for granted that everything is wired correctly.
Absolutely. It's a biological miracle that it works as often as it does.
Now, before we sign off, here's a thought to leave you with. We talked about how the brain flexes and folds, creating those gyri and sulci, the wrinkles, because it grows so much faster than the skull.
Yeah.
It's trying to pack more surface area into a very limited space.
Right. The physical limitation forces the complexity. The box is too small for the balloon, so the balloon has to wrinkle.
So here's the provocative question. If the skull didn't limit the brain, if the brain could just keep growing outward smoothly without folding, what would human consciousness look like?
That is a fascinating question.
Does the physical limitation, the box we are in, actually force the complexity that makes us intelligent? Are our thoughts a product of that compression?
Or would we just be giant-headed, smooth-brained creatures with no complex thought? It's hard to say. Pressure creates diamonds, and maybe, just maybe, cranial pressure creates intellect.
Something to think about. That's it for this deep dive into wiring the body. Remember, go read more Langman and Singh. We're just the primer.
Keep learning.
See you next time.